Hello YouTube, I am Dr. Robiul. I am a lecturer of pathology in a medical college hospital and I am making this video for my students. Hope someone finds this helpful. Okay, now today's topic is cellular adaptation. Now, cellular adaptation is a very straightforward topic of the general pathology section, but in order to understand the more difficult topics like cell injury, inflammation, you need to have a clear conception about cellular adaptation. So, what is cellular adaptation? So, since we have the term cellular in the beginning, so there is no doubt that this thing is occurring inside the cell. And before I go through the big definition of cellular adaptation, I want you to know one thing. That is, cellular adaptation is a reversible change in response to the change in environment of the cell. Okay, now what are the types of change we can have in adaptation? It can be change in the size of the cell, change in number of the cell, change in function of the cell, and even change in phenotype and metabolic activity of the cell. So whenever we're having these type of change in a cell, and whenever those changes are reversible, chances are we are talking about cellular adaptation. So I will go through the definition, but I just want to make a point that always remember cellular adaptation is a reversible change in either the size, the number, the function, the phenotype, or the metabolic activity of a cell in response to changes in the environment of the cell. Now, in your textbook, you will see a long definition of cellular adaptation, and I have also written that here. And in order to get good marks in your written examination, you have to know this definition. Okay. So, cellular adaptation, I have already mentioned, this is a reversible change. How can we define cellular adaptation? Adaptations are reversible, functional, and structural responses to severe physiological stresses and some pathological stimuli during which an altered, steady state is achieved, allowing the cell to survive and continue to function. Okay, so always remember when you try to memorize the definition, always look for keywords. So here we can see we have two keywords. The first thing is reversible. Adaptation is a reversible response. And the second important point is here the cells are achieving an altered steady state. This is a very important section of the definition. Okay, so the cells are having an altered, the cells have changed. The cells have changed either in their size or in their number, metabolic activity, phenotype. Why? To achieve a steady, a stable state so that they can continue to function. Okay, now what are the types of cellular adaptation? So always remember cellular adaptation are of four types. They are hypertrophy, hyperplasia, atrophy, and metaplasia. Now many of my students make a common mistake with the words metaplasia and metastasis. You know, in the exam hall you are writing fast, you are writing to finish in time, so you're writing hypertrophy, hyperplasia, atrophy, metastasis. No, never write metastasis. Okay, metastasis is a different topic. When we will talk about neoplasia, then we will see the term metastasis, which is seen in malignant tumors. But here the term is metaplasia. Now we will discuss each of these topics one by one, but in short, what do they mean? Okay, hyper means increased, trophy means growth. So here the cells are increasing in size. So hypertrophy, increase in cell size. Okay, the second one was hyperplasia. Hyper means increase. What does plasia stands for? Plasia stands for proliferation. So here in hyperplasia there will be increased cell proliferation or increase in cell number, right? Okay. The third one was atrophy. A means absent, trophy means growth. So atrophy means absent or decrease in the growth, leading to decrease in cell size. The last type was metaplasia, and what's happening here? One adult cell type is replaced by another adult cell type of the same tissue. For example, as I have written, you can see the normal lining epithelium of our respiratory tract is pseudo, stratified, ciliated, columnar epithelium. And what happens in a chronic smoker, those cells realize that they can no longer survive this chronic irritation and they get replaced by stratified squamous epithelium, which can better survive in this tobacco-rich environment. So that is an example of metaplasia. Okay, so now that we have discussed in short the basic concept of hypertrophy, hyperplasia, atrophy, metaplasia, you may be asking a question: when does a cell decide that the cell will increase in size, and when does a cell decide that it will increase in number? What is the concept that dictates when a cell tries to increase in size or increase in number? And now, to understand that thing, we have to take a look at the cell cycle. Okay, so I think you have read in your biochemistry classes a thing or two about cell cycle. Okay, and I have also drawn a picture of the cell cycle. Okay, can you remember? Cell cycle, it showed the different stages a cell goes through, and they were also given different names. For example, there was a M phase during which cell division occurred. Then there was a G1 phase during which what happened? The cell prepared itself for the next cell division. You know, all the organelles were getting ready for the next cell division steps. Then there was another phase called the S phase, during which we had DNA replication. Can you recall? Yes, DNA replication. What does that mean? Well, in order to uniformly distribute our genetic material in the daughter cells, first we have to double our DNA amount. If we don't double our DNA amount, what will happen? We will produce haploid cells, and that won't be good for mitosis, right? That will result in meiosis. So in all mitotic division, there was an S phase in the cell cycle during which DNA replication occurred. And after DNA replication, there was another phase called G2 phase. What happened here? Well, actually in G2 phase, these newly replicated DNA were checked for errors, you know, because if we have some error in DNA, that's not a good thing, you know. One error in your DNA can lead to a very big problem in your next generation. So those things were checked in G2 phase, and then there was again cell division, and this cycle went on continuously, and there were some cells which left this cell cycle, and we used to say they are in Z0 phase. And the point I'm trying to make here, in order to understand when a cell is dividing and increasing its number, and when a cell is not dividing, just only increasing its size, depends on the type of the cell. You know, cells that are in the G0 phase, they can no longer divide. They are known as permanent cell. And can you give me examples of permanent cells? Yes, permanent cells were our neurons. Then we had our skeletal muscle, and also we had our cardiac muscle, right? Okay, so say we are increasing workload in skeletal muscle or cardiac muscle. What will happen to them? They need to cope with the increased workload, right? But they cannot divide. So the only way for them to adapt is increase in cell size, that is hypertrophy. But remember, there are other cells that can divide, and we can classify those cells able to divide into two ways. One is called stable cell, and one is known as labile cells. Okay, the stable cells, they can divide, but they divide only when needed. For example, in the liver we have hepatocyte. Are they dividing all the time? No, but they can divide. For example, if a patient had partial hepatectomy, part of his liver has been cut out, then the remaining liver cells, they can increase in number by dividing, and they are an example of a stable cell. And the third group, according to the cell division, is the labile cells. The labile cells are continuously dividing, you know. For example, the lining epithelium of our gastrointestinal tract, the epithelium of our skin. These are always dividing, and they are called labile cells. So labile cells will have a more preference for hyperplasia because they are very good at cell division. Okay, so always keep this in mind, that thing about cell cycle, when you ask the question when a cell is choosing to increase its size and when it's choosing to increase its number. Okay, so now let's go deeper into the subject, and we will start our discussion of the cellular adaptation with the first one, which was hypertrophy. Hypertrophy refers to an increase in the cell size, resulting in an increase in the size of organ. Okay, now hypertrophy can be of two types: physiological hypertrophy and pathological hypertrophy. So can you give me an example of physiological hypertrophy? Yes, very simple. The bulging muscles of bodybuilders who are engaged in, you know, exercise like pumping iron. There, what's happening? There, the muscle fibers are not increasing in their number; they're increasing in their size. For example, if say this was a muscle fiber of a person before he went to the gym for exercise, and a few months later, what will happen to that muscle fiber? The muscle fiber will be hypertrophied, and here the number of muscle cells did not increase, only the size of those cells have increased, and that's giving it an overall appearance of increase in the muscle bulk. Now, another example of physiological hypertrophy we can see is in the pregnant uterus. Okay, and there we are having hypertrophy of the muscles. But also remember one thing: in the uterus we can have both hypertrophy as well as hyperplasia. Now, what are the examples of pathological hypertrophy? Okay, now pathological hypertrophy here we are increasing our cell size due to some abnormality. The most common example of pathological hypertrophy is left ventricular hypertrophy. I can write LVH due to systemic hypertension. Okay, now why is this happening? Let's go to an explanation. So I have drawn a very simple diagrammatic picture of a heart. Okay, now why would we have hypertension? That's the first question. You know, there are different reasons for hypertension. Say, for example, this was a blood vessel. You can see I'm drawing the lining endothelium. Okay, these are the nucleus. The dots are the nucleus of the endothelium. And say for some reason that patient had high LDL, low density lipoprotein, in his blood. Maybe he takes a lot of, rich food. Okay, then those LDLs damaged this endothelium and went beneath the endothelium, subendothelial space, and there those lipids accumulate. Okay, later what happened? In the blood we have different types of cell. One of those cells are macrophage. They came, they saw the damage, and they went through the gap beneath the endothelium to engulf those lipid particles. But ultimately they die. And what happens, we can see later that lipid particles are accumulating beneath the endothelium, and this is known as atherosclerosis, right? So atherosclerosis, what's it's doing on the blood vessel? See, it's narrowing. Look at the diameter of the blood vessel here, and look at the diameter here. The blood vessel has gone narrow, and whenever a blood vessel, say an artery, gets narrow, it increases the pressure, it increases the blood pressure, and that increases the afterload, the pressure against which the left ventricle has to pump out, So what will happen? Since the afterload, when we are increasing the afterload of the heart will try to adapt. And how will the heart adapt? It will increase its muscle mass in the left ventricle. Say now it will become more thick. The left ventricle will have more thicker muscle. Okay, and that will lead to left ventricular hypertrophy. So this is an example of pathological hypertrophy. Okay, so this is in short about hypertrophy. Okay, the next question is, what is the mechanism of hypertrophy? Now this is a very molecular level question. What is the mechanism of hypertrophy? What is happening when the heart is having to work against increased afterload? What is providing the stimulus for hypertrophy? So there are different types of hypotheses, and the main things that you need to know is there are mainly three mechanisms. One is mechanical sensor mediated. For example, in our heart we have some mechanical sensor, for example integrin, and those things are triggered by increased workload, and then they do what? They increase transcription. What is transcription? Transcription, they increase transcription means they are making RNA from DNA, and those RNA, say mRNA, messenger RNA, they contains blueprints for making different types of protein. Okay, so mechanical sensors are getting stimulated, they are releasing transcription factor, which is increasing transcription, and what's happening? Different types of proteins are then forming. So this is one way hypertrophy can occur. There are two other stimulants in your textbook. One is different types of drugs and agonists, say for example alpha adrenergic hormones. They can also act as stimulants and increase release of transcription factor and then increase protein synthesis, leading to hypertrophy. Okay, and the third way hypertrophy can occur is by some growth factors. Growth factors, can you name some growth factors? Yes, they are like IGF, insulin-like growth factor 1. We also have other growth factors like TGF, etc. So they will also increase transcription factor in the cells that we want to increase in size. That will increase protein synthesis. For example, here there will be increased myofilament synthesis in the myocardium, and that will lead to hypertrophy. Okay, so the point I'm trying to make here, hypertrophy can occur due to some mechanical sensor, due to some agonist, or due to some growth factor, and they are all increasing transcription factor formation, which is leading to increased protein synthesis and causing the hypertrophy. Okay, so this is the main thing about hypertrophy. The next point was hyperplasia. As I have said earlier, hyperplasia means increase in the number of the cell, which leads to increase in the organ or tissue size. And as like hypertrophy, hyperplasia can be of two types: physiological hyperplasia and pathological hyperplasia. Okay, so let's first begin with physiological hyperplasia. Now, physiological hyperplasia can occur by two ways. One is known as hormonal and the other one is compensatory hyperplasia. Okay, compensatory. Now, can you give me an example where we are having increase in number of cell due to hormonal reasons? Well, hormonal hyperplasia is well illustrated by proliferation of glandular epithelium in the female breast during puberty. So one example of hormonal hyperplasia is female breast, which is enlarging in size due to glandular cell proliferation during puberty. And also, I had said earlier, the hyperplasia of the pregnant uterus is also another example of hormonal hyperplasia. But always remember, in uterus we are having both hypertrophy and hyperplasia. Okay, now can you give me an example of compensatory hyperplasia? Now, compensatory hyperplasia occurs in the liver, and also in your textbook you will see a nice story about Prometheus. Yeah? Can you recall? Now, who was Prometheus? No, this is not the spaceship you saw in the movie Prometheus. No, it's not that. Well, Prometheus was someone who stole the secret of fire from the Greek gods. And then what happened? The Greek gods went very angry and they decided to punish Prometheus. And how did they punish Prometheus? They chained him with a mountain, and every day his liver was devoured daily by eagles. And what happened in the night time? The liver grew back again. Okay, so every day the liver is getting devoured by the eagles, and at night time it's growing back again. So the point I'm trying to make here is liver can increase its cell number when needed, and that is compensatory hyperplasia. Next is pathological hyperplasia. Now, what are the causes or examples of pathological hyperplasia? As you can see in your textbook, examples are hyperplasia of the endometrial glands due to some abnormal increase in the estrogen level. Second cause is benign prostatic hyperplasia, and even in some viral infection, say for example in the skin warts of papillomavirus, there also we can see hyperplasia. Now I will explain the first one, why we can have hyperplasia of the endometrial glands. Can you recall, in the female menstrual cycle, 4 hormones are responsible. Can you recall? The hormones were follicle stimulating hormone and luteinizing hormone. These are released from the anterior pituitary gland, and there was also 2 more hormones, estrogen and, in fact, the subtype of the estrogen was estradiol, and there was also progesterone. Can you recall? Okay, so what happened? These hormones dictate the menstrual cycle. First, follicle stimulating hormone used to stimulate development of one follicle in the ovary. One follicle got bigger in size than the other, it got bigger and bigger, and then it started releasing estrogen. Okay, and that estrogen had an effect on LH. As you can see, this is the level of LH, this is progesterone, this is estrogen. As estrogen level was rising, it used to stimulate the release of more and more luteinizing hormone, and then there was a sudden surge in the luteinizing hormone. This is the luteinizing hormone surge, and that led to rupture of the ovum and ovulation occurred. And just after ovulation, we can see that there was a drop in the estrogen level. You see, after ovulation, the estrogen level suddenly drops, and then the luteal phase began, and then there was increased secretion of the progesterone. Okay, so this is the normal physiology of menstrual cycle. Now what will happen if, due to some abnormality, say we have a tumor or something that is increasing the estrogen level? This entire balance in the cycle will be disrupted. There will be more and more proliferation of the cells, and that will lead to pathological hyperplastic proliferation of the endometrial glands. And when those things later slough out, that will lead to dysfunctional uterine bleeding. Okay, the next example, as I have written, was benign hyperplasia of the prostate, or benign prostatic hyperplasia. And what happens here, for some reason, the patient has increased level of androgen, and that stimulates the proliferation of the prostatic gland. And we know that part of our urethra resides inside the prostate, so the prostatic part of our urethra will get compressed, and those patients will give us complaints that they have difficulty in initiating and stopping urination. So whenever you hear those type of complaints from a patient, and if he is an older male, do think that that patient can have possibility of benign prostatic hyperplasia. Okay, and the third one was papillomaviral skin wart. The last point I would like to mention about hyperplasia is the mechanism of hyperplasia. So hyperplasia mechanism, 2 ways. One is growth factor driven proliferation of mature cells, and the second mechanism is formation of new cells from stem cell. That means the second mechanism is stem cell driven. Now, you may be asking me when a hyperplasia is occurring due to proliferation of mature cells, and when the tissue decides that it has to make new cells from the stem cells themselves. Okay, so let me give you an example. Say a patient has undergone partial hepatectomy. That means part of his liver has been cut out, but some part is still inside the patient. And in the remaining part of the liver, the cells that survived the hepatectomy, those cells have receptors on their surface for growth factor. And what happens after partial hepatectomy? Liver produces and releases some growth factor. Those growth factors binds with the surface receptor on the survived hepatocyte, and they activate the signaling pathways that stimulates cell proliferation. So this is an example of growth factor driven proliferation of the mature cells. And another example I will give you is stem cell driven proliferation of the cells. Okay, so suppose the liver was infected by some virus. The patient had viral hepatitis, and the viral hepatitis was so severe that most of the hepatocyte lost their regenerating capacity. So what will they do? Then the liver will decide that we have to make newer cells from intrahepatic stem cells. So in those cases, there will be newer cells produced from not the mature cells, but from the intrahepatic stem cells themselves. So this is the basic two main mechanism of hyperplasia. The third topic of cellular adaptation was atrophy. Like I said, A means absent, trophy means growth. So here we are having reduced size of an organ or tissue resulting from a decrease in the cell size and number. Okay, so both can result in atrophy. So what are the causes of atrophy? Like the previous ones, we also have two groups here. The first one is physiological, and the second group was pathological, right? Okay, so what are the examples of physiological atrophy? Well, there are a lot of examples of physiological atrophy. Say, for example, in our intrauterine life, we had a lot of structures that are no longer seen in the adult life. For example, notochord, thyroglossal duct, What happened to those structures? Those structures were important in the intrauterine life, in the embryonic life, but not important after birth. So those things undergo atrophy during fetal development. Okay, so that is an example of physiological atrophy. Another example is the decrease in the uterus size shortly after parturition. Shortly after childbirth, the uterus starts to shrink its size, and that's also an example of physiological atrophy. Okay, and you will have a lot of questions from atrophy, so you have to remember all the causes. Now, pathological causes of atrophy. In a textbook, you will see a long list, and I have also written that here. So let's discuss these things one by one. Causes of pathological atrophy. The first one is decreased workload. So say, for example, a patient had a bone fracture. Okay, the patient had a bone fracture, so he went to the doctor. They gave him a plaster cast, and the patient has been advised to complete bed rest, So his skeletal muscles that are involved in the fractured bone, they are now resting, they are not getting used. So what will happen? Those skeletal muscle cells, since they are not using themselves anymore, so they will decrease in size, and that is an example of disuse atrophy or atrophy due to decreased workload. Okay, the second cause of pathological atrophy is decreased innervation. Okay, so can you recall the movie Avatar? Yeah, have you seen that? I'm sure you have seen that movie. Okay, so have you looked at the leg of the hero in the movie? The patient, the hero of the movie was paralyzed. You know, his legs were paralyzed, and therefore, if you recall, his legs, the muscles of his legs were atrophied. You know, so that's an example of disuse, denervation atrophy due to loss of innervation. Since there is loss of innervation, the skeletal muscles are not getting used, and since the normal metabolism and function of a skeletal muscle are dependent on its nerve supply, so when there is no nerve supply, those muscles are getting shorter and resulting in denervation atrophy. Okay, the third cause is diminished blood supply. You know, say for example, the patient had a block, an occlusion in his artery, and then what will happen? The area in which that artery supplied nutrient, those area will get less and less blood supply, less and less nutrition, and then they will diminish in size. An example of diminished blood supply atrophy, you can see in your textbook there is an image that's occurring in the brain of people in old age due to reduced blood supply in the brain. And when there is reduced blood supply in the brain, there are some changes occurring in the brain. Okay, so that is an example of senile atrophy of the brain. The next point, the next cause of pathological atrophy is inadequate nutrient. Okay, so what happens when, say for example, a patient has marasmus or some other protein-calorie malnutrition? Okay, then first our body will burn out all his carbohydrate, all his fats, you know, as a source of energy when there is nothing else to burn except protein. Thus, our body will try to make energy from the protein also, and that will lead to wasting of the muscle, and that is an example of atrophy due to decreased nutrient. Okay, the next cause of pathological atrophy was due to loss of endocrine stimulation. For example, many tissues in our body, for example, the female breast, the female reproductive organ, etc., are dependent on endocrine stimulation. Okay, so when we are stopping those endocrine stimulation, those organs will become atrophied. Okay, the last one is pressure atrophy, and the idea of this atrophy is, say for example, a tumor, it may be benign, it may be malignant, is growing. And when a tumor, a space-occupying tumor is growing, it's compressing its surrounding structure. So if there were cells here, say for example these are cells and this is a tumor. Okay, so when the tumor is growing, the tumor is compressing these cells, and then in order to survive, those cells will decide that if they shrink in size, they can better withstand the pressure. So that's an example of pressure atrophy. Another example of pressure atrophy is in cystic fibrosis. Okay, now what is cystic fibrosis? Cystic fibrosis is a genetic disorder where we are having defective chloride channel formation. Okay, the chloride channels are so defective that normally chloride channels are one type of protein. Okay, all channels are in fact proteins. So chloride channel, that's also a protein, but that was not created properly. There was misfolding of the chloride channel protein due to mutation in CFTR gene. Okay, so what happens? Those misfolded proteins fail to go, say for example this was protein chloride channel, to go from the cytoplasm to the cell surface, So those chloride channels cannot work, and chloride cannot be secreted in the secretion. So say for example this was a gland. Okay, this is a diagrammatic image of a gland. These are all columnar cells. They are secreting different types of glandular secretion, but there is defective chloride channel, so less and less chloride is produced and secreted by these cells. So the secretion of these glands become very thick, so thick that they block the duct of the gland. And when the duct of the gland is blocked, these secretions have nowhere to go, so they create a very high pressure inside the glandular epithelium, and that pressure leads to atrophy of those glandular cells, and this is also an example of pressure atrophy. So here are some images from your textbook, and this is an example of senile atrophy of the brain. As you can see, we have two images here. On the left we have a normal healthy brain. You can see on the right side of this image, the meninges, the covering has been removed, and on the left side we still have the meninges with all those vessels. Those are all vessels on the meninges. So here the meninges has been stripped off, and you can see these are the elevated ones are the gyrus, the gyri of the brain, and the grooves are known as the sulcus. So this is the normal brain. And on the next image, what has happened here? Can you see this image? Here also on the left side we still have the covering meninges, but on the right side you can see that the covering has been removed. And look the difference. Notice the difference between this sulcus and these sulcus. You can see the sulcus here have widened and the gyrus have narrowed, and this is a characteristic feature of senile atrophy. The last point about atrophy is the mechanism of atrophy, and there are actually two mechanisms. The main idea, the main way atrophy results is by decreasing protein synthesis and increasing protein degradation in the cell, and that thing can happen in two ways. So one of the ways is, like I said, ubiquitin proteasome pathway, and the second way, the second mechanism of atrophy is by self-eating process known as autophagy. Okay, so what is the ubiquitin proteasome pathway? When a cell is having or facing nutrient deficiency, that leads to activation of an enzyme known as ubiquitin ligase. Okay, so what does this enzyme then do? That thing then activates small peptide molecules known as ubiquitin, and those ubiquitin proteins target some cellular protein and binds with those proteins. And ubiquitin is like a tag. When they are bound with certain proteins, those proteins can be targeted by proteasomes for degradation. So when, say for example, this was a protein, it is now coated with ubiquitin. These dot lines I am drawing are, say, ubiquitin. Now this thing can be recognized by proteasome, and they will come engulf these ubiquitin laden protein molecule, and then it will degrade that protein. So as protein will be degraded, the cell will lose its proteins, and it will decrease in size. The second way by which atrophy can occur is self-eating of its own organelles. When the cell is facing severe nutrition deficiency, sometimes it will try to digest its own components. So in those cases, there will be formation of some autophagic vacuoles. That means there will be some vacuoles formed, and those vacuoles will contain fragments of its own organelles, and then those autophagic vacuoles will bind with lysosome, and then the lysosome enzyme will degrade those things. So this is the mechanism of atrophy. Okay, so the last topic of our cellular adaptation was metaplasia. So what do we mean by metaplasia? Metaplasia, like I said, is a type of adaptation in which one adult cell type is replaced by another adult cell type of the same tissue. So there are mainly two types of metaplasia: epithelial metaplasia and connective tissue metaplasia. First, let's talk about the epithelial tissue metaplasia. Okay, so we know there are different types of epithelial tissue. We have squamous cell, we have columnar cell, we have cuboidal cell, etc. So the most common type of epithelial metaplasia is stratified squamous epithelium metaplasia. That is, different types of epithelial cells will be replaced by much more stronger stratified squamous epithelial cells. Say, for example, in our respiratory system, what was the lining of our stratified? Can you recall? Yes, pseudo, stratified, ciliated, columnar epithelium. What does these words mean? Pseudo means false. Stratified means multiple layers. So, if I draw some pseudo stratified epithelium, say this is one cell, this is another cell, this is another cell. Okay, these are the nucleus of those cells. So notice one thing: all the cells are touching the basement membrane, right? So in reality there is just only one layer, right? So this is not a stratified epithelium. However, notice the height of individual cells. Since the height of these individual cells are not equal, and since when we look the cells under microscope, we actually see the nucleus more prominently. So look here, actually it seems as if we have nucleus in two layers, so that's giving it a false stratified appearance, and from there the name pseudo stratified came from. But actually all are columnar epithelium. Okay, the picture is not perfect. Columnar epithelium, the nucleus should be near the basal hub, and moreover these are ciliated epithelium. So let's draw some cilia. What's the function of the cilia in the respiratory tract? They are continuously beating upwards, and all the dust particles that we inhale, almost all of those dust particles are propelled backwards, so that our alveoli are always dust free and clean. And they do that thing, they achieve that thing by the synchronized beating of the cilia, like the Mexican wave you see in the stadiums. Okay, so this is normal pseudo stratified ciliated columnar epithelium. But what happens in case of chronic smoker? tobacco is irritating these epithelium, and then those epithelium changes into something much more resilient. Let's draw a real squamous epithelium. If you can see, here we have multiple layers of cells. Can you see? 1, 2, I have drawn 3 layers of cells, and look, not all the cells are touching the basement membrane. So this is a real stratified squamous epithelium, and this was pseudo stratified. So what are the other examples of squamous metaplasia? The second one is columnar to squamous epithelium. There are lots of examples. For example, stones in excretory duct of glands, like salivary gland, pancreas, bile ducts may also cause replacement of the normal columnar epithelium by stratified squamous epithelium. Vitamin A deficiency also stimulates replacement of normal cells by stratified squamous epithelium. So these are the examples of squamous metaplasia. Next we have columnar metaplasia, and here we will have a very important topic known as Barrett's esophagus. Okay, this is very, very important for your exam. So what's happening here? Say a patient has GERD. What does GERD means? Yes, gastroesophageal reflux disease. Acid content from his stomach is backflowing into the esophagus. Now, what was the normal lining of the esophagus? The normal lining of the esophagus was squamous epithelium, right? But when acid content is coming to the esophagus is not designed to manage hydrochloric acid. So when it sees this hydrochloric acid, it tries to adapt, and it replaces itself by much more resilient intestinal-like columnar epithelium cells. So this is an example of columnar metaplasia. Now these were all epithelial metaplasia. There can also be connective tissue metaplasia, where one connective tissue cell is replaced by another connective tissue cell. Say, for example, if someone gets a trauma in his muscle, sometimes there is bone formation inside the muscle, although that's not very common, but sometimes there can be bone formation inside your muscle cells, and that is known as myositis ossificans. So that is an example of connective tissue metaplasia. The last point I would like to say about metaplasia is the mechanism of metaplasia. Now, how does metaplasia occurs? Okay, remember, we are not changing. In the definition I said replaced. Okay, replaced. Squamous cell is not getting converted to columnar cell, or columnar cell are not getting converted to squamous cell. They are replaced. Newer cells are forming, and how this thing is occurring, the mechanism is reprogramming of the stem cells. This is very important. You have to know this. The mechanism of metaplasia is reprogramming of stem cells. So, stem cells, they are, pluripotent. They have a lot of potential. They can make any type of cell. That's why now we are researching so much on stem cells, So, when we can reprogram a stem cell, so they can think like we are facing more hydrochloric acid in case of Barrett's esophagus. So why not make intestinal-like epithelium instead of making squamous cell? And that's the mechanism of metaplasia. Now, in your textbook, you will see a term called double-edged sword. Metaplasia is sometimes called double-edged sword. Now, what's a double-edged sword? And it means a sword that has sharpness in both sides. And that has some advantage and disadvantage in fighting. If you are a sword fighter, you can hurt yourself also. Similarly, metaplasia has both beneficial and harmful effect. What's the beneficial effect of metaplasia? The metaplastic cells are much more resilient to the stress. So these cells can survive longer. But the backside to this is they sometimes lose their vital important functions. Say for example, when we are converting the respiratory epithelium into stratified squamous epithelium, that can better withstand the harmful effect of tobacco. But however, they will lose mucus production capability, so that's not a good thing. And another disadvantage of metaplasia is this is a fertile soil for malignancy. Sometimes when we are reprogramming stem cells, something can go wrong, and if the chronic irritation persists, say for example a patient had GERD and that caused squamous epithelium of the esophagus to replace by some much more resilient tissue, but those causes of GERD were not corrected, then ultimately the stem cells can do some mistake when they are trying to reprogram themselves, and that can lead to malignancy. Okay, so this is in short about the 4 types of adaptation. I hope you learned the basic concept of adaptation, and I also recommend you to go through your textbooks to see much more images. Okay, that's all for today. Take care.